I'm so glad that you're all here. I'm really excited. It's my distinct honor and privilege to introduce Matt. I just asked him, what do you want me to say about you? And he said, as little as possible. So here's Matt. No,
Thank you.
Okay, so he's a Sherman Fairchild professor of neuroscience at MIT. And when they talk about Johnson potential speakers at the beginning of the year, I thought of Matt right away because, you'll find out of his cool research, but he's also a passionate teacher. But today I also found out that when he was at RPI as an undergrad, not only he studied electrical engineering, but he also majored in philosophy.
Minored. And psychology.
Psychology. So I think it really fits into our ethos of liberal art, engineering, all together. So I'm really excited that Matt is here. He earned his PhD from Caltech in I won't say what year, but I've known Matt for over 10 years, and I'm so grateful that he's here. Here's Matt.
Yeah, great to be here. Thank you. So I want to keep it short because I know I have way too many slides, and I'm really going to try to get through as much, hopefully, all of this as I can. And my objective is to really sort of inspire what I've heard sort of this engineer-scientist view. I was trained as an engineer. I was really intrigued with the brain, and I've always sort of approached the study of the brain with the sort of the eye of an engineer. What is it we need to understand about complex biological systems if we were to try to, artificially synthesize an intelligence? And so I like to try to point out principles that I have derived from the study of the brain that I think are relevant to our understanding of what it means to think, to understand. What is it the brain does that current AIs perhaps don't do and we might be able to do better? And I'm going to start that, the journey. Let me see if I can, so I can turn this thing off. So I don't want to go back too much in the history of it, but I kind of, at Caltech, I was studying the olfactory system, I was building neural models, and I realized I need to know more about what actual neural populations in the brain do. And I need to understand this in animals that are actually engaged in a naturalistic behavior. Now, how could I actually do that? Well, it turned out at that time, and this was around in the early 1990s, around 1991, there was a lab at the University of Colorado, run by Bruce McNaughton, who was studying an interesting phenomenon. That was the phenomenon that had been first discovered by John O'Keefe in the early 1970s. And that is implanting electrodes into this region of the brain, the hippocampus. And this is shown here in cross-section. Here's the hippocampus lying deep in the medial temporal lobes. And when O'Keefe recorded neurons in the hippocampus, what he discovered in the early 1970s was that these cells, when animals were allowed out of the head bars that they had normally been placed in to study hippocampal function and associative memory, and was simply allowed to move freely on this tabletop, that the cells exhibited spatial receptive fields, what he termed place fields. He called the cells place cells. And what Bruce McNaughton was doing at the time was trying to record place cells in animals, in freely behaving animals, using some technologies that involve like multiple wire electrodes. And so I kind of came to the lab thinking, I want to record neural ensembles. The hippocampus, it's interesting. I don't know exactly what it does, but he's doing it in freely behaving animals. As an engineer, it's like, this is great. Neural ensembles, freely behaving animals. The hippocampus is involved in memory. So that was the other piece, a memory structure that you can access and do electrophysiology in that had a compelling behavioral correlate. And part of the motivation in studying the hippocampus was that this work linking the hippocampus in humans to episodic memory. Of course, the seminal case of the patient HM, or Henry Meliason, he's recently passed and had been studied for many years by Suzanne Corkin at MIT, first studied by Brenda Milner. And, HM underwent a procedure involving the bilateral resection of the medial temporal lobes, took out his medial temporal lobes for intractable epilepsy, lost the ability to form any new memories of experience. Damage to the hippocampus in animal models in rodents impairs spatial navigation, and also in humans. So like, what's the connection between episodic memory, memory of experience, and memory of navigation in space? And so there's a very sort of simple working hypothesis that I like to, to put forward, and that is, in both of these cases, there's this necessity to capture, both encode and then process information about temporal sequential information. So things occurring in time. And one of the sort of the fundamental reasons that this would be fundamental is that one of the imperatives of the brain is to be able to predict things in the future. Based on past experience, what's going to happen in the future? And the future is not always known. It may occur in novel contexts. So how do I make adaptive predictions in the future? I need to understand if, in a fundamental level, temporal order so I can infer causality. It's not just knowing what happened after what, it's knowing why things happened after, one after the other. And so that's sort of going from what to why causality. And what I like to point out is sort of the third level. Once we understand what happened, once we kind of infer why it happened, now we need to convert that into sort of a generative form where we can sort of imagine or execute actions that will cause it to happen. How do I make this happen? What happened? Why did it happen? How can I make this happen again? And so this is going to be the basic process that I'm going to sort of put forward as being fundamentally dependent on our ability to capture and process temporal sequential information. And this is why the hippocampus would be fundamentally important. Not necessarily because it's uniquely capable of processing temporal sequential information, but because of where it actually lies in the brain hierarchy. If I were to sort of put forward a kind of a hierarchical chart starting from like sensory systems, information comes into the brain, sensory cortical areas, and then you see this hierarchy converges into higher level associative areas, and then, increasingly converges till it gets to the top of the hierarchy. And there are two structures that classically are placed at the top of that hierarchy. The one that we believe places us at the pinnacle of evolutionary intelligence, that's the prefrontal cortex. Prefrontal cortex, highly evolved in humans, lets us make great or not so great decisions, but we make decisions with it nonetheless. The other structure that lies at about the same level is the hippocampus. So the hippocampal lies at the point, it's able to integrate information from across the brain and then perform this kind of critical function of encoding and evaluating temporal sequential information. So how does it actually do this? Well, the first step is to understand, what is this encoding and how does it manifest in the level of neural circuits? And so very simple technology is placing wires, doing extracellular electrophysiology, placing wires, insulated wires, about 35 microns across, with the tips exposed so you can record the electric fields generated by action potential or spiking events in neurons. And one of the things that I did when I went to Bruce McNaughton's lab that was a sort of a modest innovation was, instead of putting in single wires, you put in small bundles of wires that have 4 contacts closely spaced. What this allows you to do is to kind of triangulate the location of the sources based on just the kind of the amplitude attenuation that comes as a function of distance. And if you put a bunch of those into the brain using a little microdrive device like this, where each individual electrode or tetrode, as we refer to them, 4-wire electrodes, each can be individually advanced. You put them into the cell body layer like this. You put them next to cells. It's like this is a cartoon. Just they sort of look like real cells. But this is approximately the dimensions, the proper dimensions, about 35 microns across. You can record the activity from multiple neurons at single sites. Put in a bunch of these. You can now record hundreds of neurons. And so we're going to look at a population of neurons in a freely behaving animal. And this just shows the raw activity. These individual points are individual action potentials displayed in an amplitude space. So this is like recording an action potential generated by a single cell recorded at 4 contacts. You're looking at 2 of these channels or contacts. And you see that spikes that show up here, individual points here that are large on channel 1 and small on channel 2, mean that the source of the cell is close to channel 1 and far from channel 2. So you can do like a spatial triangulation, pull out a bunch of cells, and then ask the question, what do these cells do in an animal moving in space? So this is just an illustration of the phenomena that John O'Keefe had discovered and was given the Nobel Prize for in 2015, and that is the property of place-specific firing, place cells. So these are place cells. Each of these points is an action potential, a spike generated by a single cell that's color-coded. So here's a yellow cell, red cell, blue cell, green cell. And these are like two, we refer to them as mazes. They're just little linear tracks, about 1.5 meters here. Animal starts at one end, runs down the track, goes to this end, gets some food, runs back and forth. So they're just kind of alternating on this linear track. And what you see is that different cells fire in different portions of the maze. Yellow cell fires this part of the track, red cell here, green cell here. Animal turns around, the blue cell fires. So a couple of things about this. You put them onto a different track, different cells fire in different configurations. So the pattern of activity of individual cells changes with context. And what's important to point out, and the reason that this technique really kind of works at all, you put the electrodes in to the hippocampus. They're not placed to kind of optimize the receptive field, the detection of receptive fields, as you might in the visual cortex. For instance, if I wanted to record cells that respond to this part of the visual field, I have to put them in the right place in the visual cortex where the receptive fields. In the hippocampus, pretty much any of the cells anywhere are going to be capable of expressing place-specific firing. And in any given environment, put the animal on this track, 30 to 50% of the hippocampal neurons are going to have place fields. So this tells you a couple of things. One, good place to record because you're pretty much guaranteed to get a lot of cells in the hippocampus. The second thing is no individual cell actually carries sufficient information to distinguish a particular context. And that is, I can't tell, where I am in a given maze with just a single cell, because this yellow cell, sure, it'll fire here, but if I put it into another maze, it's just as likely, with about a 30 to 50% draw chance, of firing there as well. So the thing that uniquely defines both a context and a location is the configuration of cells, the pattern across a large ensemble of cells. And at any given point, at any given time, roughly 3 to 5% of all the cells in the hippocampus are active, about 100,000 cells. So you think, like the vector that defines a unique location, you could say experience, is a pattern of about 5,000 cells out of about 100,000 cells. It's that pattern that's really unique. So we're going to look at, I'm only showing you, let's say 6 cells, but we're actually recording about 100, and they're going to be at any given location. There's going to be a pattern of, a small handful of cells. And so it's that configuration of cells that we'll use to uniquely define the location. And this just kind of illustrates, this is a movie, and I just use this to, see if I can play this, sort of illustrate the fact that this spatial firing it's something that doesn't just emerge after a lot of post-processing. It's evident in the raw data. So this is activity taken right from the animal's brain. You're seeing this blue cell fire here, for instance. That's kind of weird. The point is weird. Blue cell firing here, light blue cell firing here. But now you notice the animal stopped. And if you listen really carefully, you can hear that something actually changed. When the animal moves, if you listen, there's going to be a rhythmic modulation. It's going to be this kind of, if you listen, and they'll stop. Can anybody hear it out there? The rhythmic modulation? And if you listen to the stuff long enough, it just jumps right out at you. When the animal moves, there's a rhythmic modulation at about 10 hertz known as the theta rhythm. When the animal stops, that rhythm goes away and is replaced by this aperiodic bursting. I won't play it again, but if you can both see the change in activity, the way the cells are active, and the background dynamics also change. You go from rhythms during active locomotion, and what I would refer to as like active encoding. You're taking in information about the world. When the animal stops, is inattentive, is engaged in behavior like grooming that doesn't require any kind of external environmental information, the rhythm goes away and is replaced by this kind of aperiodic bursting activity, in which when you look at the populations, it exhibits what are referred to as these sharp wave ripple events. I'll describe those a little bit later. But there's like 2 modes: rhythmic, place cell encoding mode, and then this aperiodic bursting, offline inattentive mode, where you see lots of cells firing in what appear to be a non-spatial fashion. Now, this is just like the raw data, but if I now take this data, take the spiking information, and then use a simple Bayesian decoding approach, which simply says, if I look at any given time, I ask which cells are firing, and then I compute the conditional probability of being at a given location, any of the given locations on this track, given the particular cells that are firing. And I just compute a probability at every location, probability the animal's here, given that the blue cell is firing. What about here, 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 every location? And then I display that probability as a little triangle. The size of the triangle is the probability. So we're going to do like every 100 milliseconds, I ask which cells are firing, and then display the conditional probability of position as a function of that firing. And what you see here, this is the ensemble decoding. I'm going to do that for a bunch of cells that are firing at a given time. And so this is what it looks like. Activity, triangle. Now, mind you, there's a blue triangle at every single location in here. It's just that here, they're so small, it doesn't show up as anything, right? Probability is 0. And so what you see is when the animal's moving, the neural code in the hippocampus tracks the animal. Not that surprising, place cells would have expected that. But notice what happens when the animal stops. So the animal's stopping, it's like, man, I can't decode this anymore. It's not really. Then suddenly you'll see bursts of activity, and you notice that triangles appear. They appear at remote locations. Now we'll come back and revisit that. What you see is these two modes, the rhythmic dynamic theta mode corresponds to, let's say, animal's current location, although we'll get into that in a little bit more detail. It's not just current location. It's actually a current local trajectory. And then when the animal stops, you get remote reactivation in temporally compressed form of distant trajectories. And interestingly, that kind of bursting activity, the remote reactivation that occurs during inattentiveness, also reappears during another state, and that's the state of sleep. So you can think of sleep as kind of a period of extended, inattentive, quiet wakefulness, right? And from the hippocampus perspective, they're basically indistinguishable. So now the animal's taken off the apparatus. He's not on the maze anymore. He's placed in a little sleeping area, and you're recording the same activity in the hippocampus. And when you decode that, what you find is during sleep, you get these little bursts. And during these bursts, you see that there's a kind of expression of a recapitulation of a spatial trajectory. Now, you might have noticed the triangles that I showed you were not just the probability, but they also had a little direction. And the direction was in the direction of motion. And that is that the cells that I showed you, place cells on those tracks, don't simply fire as a function of position, but also heading direction, that's the direction of motion. And you can also think of direction of motion as not just direction of motion, but it's the sequential dependence of firing. That is cell 1 firing, followed by cell 2. And so that the hippocampal code carries information about location and potential trajectory. So what you're seeing there is that triangle that points this way. Well, the time series was not in the direction of the triangle, which is how the animal would actually be moving. It was in the reverse direction. And that is that these reactivated events can occur in time reverse form, as though the animal were thinking backward in time. So these are just some of the basic phenomena. But I kind of want to go in a little more detail. What did they actually look like, and how are they coordinated with other structures in the brain to inform us as to what their function might actually be with respect to memory? And so, again, just kind of going back to this idea of the hippocampus having these two modes, the rhythmic theta mode that's tied to active locomotion, also associated with, for instance, in non-human primates and in humans, can be associated with sensory exploration. So you get these rhythms when you're actively engaged in exploring the environment. If you're moving, you get the rhythm, but also if you're kind of visually exploring in humans and non-human primates, you can also get this rhythm. So you can think of this rhythmic, the theta rhythmic mode, as being this, what I refer to as the online mode, taking in information. And very quickly, if an animal stops moving, you get these kind of large, these bursting events, which I refer to as sharp wave ripples. They're referred to as sharp waves because in this measurement of the local field potential, or like the kind of the macroscopic population electric field associated with like the EEG, what you see are these large deflections. What this means is that there's a lot of synchronous activity that's coming into the hippocampus at that time. And this was a figure published by Yuri Busaki to illustrate this phenomenon. And what you see here is kind of a low temporal resolution figure, and it doesn't carry, what I'll show you later, is this addition of these high-frequency oscillations that ride on top of these sharp waves, hence the term sharp wave ripples. Ripples are the high-frequency oscillations on top of this. So we've got rhythms, theta, and we got sharp wave ripples. So let's kind of go through both of these. What goes on with these theta oscillations? Well, you can say the theta oscillation, brain rhythms are, they're kind of everywhere. And you could say they just sort of kind of express the active mode of the brain. But another property of rhythms is that they're able to temporally synchronize activity. And that is the spiking activity that occurs in the hippocampus is locked or phase locked to this rhythm. And that's illustrated here. So here, in red, you see a recording of the local field potential. The local field potential is largely generated by current dipoles that result from synaptic inputs. So you have synaptic currents, they flow into cells, they generate electric fields. So you can kind of think of these rhythms as reflecting, this is how the inputs are actually being modulated. The spiking, of course, is the output. And so what you see here is that these rhythms, the spiking, for instance here in the hippocampus, the spikes are kind of phase locked around the peaks of these rhythms. And you say that's not that surprising, right? If the inputs, this is like the inputs, you spike when you get a lot of input. You get inputs, you generate spikes. Well, and this property was known. Well, John O'Keefe made another very seminal discovery and observation about the hippocampus in the early 1990s. Again, place cells in the 70s, this property of spiking in the hippocampus in the 1990s. And what he noted was, you look at this and you say, yeah, they're kind of phase locked. The spikes occur near the peak. But what he noticed was, as an animal, this is in time, but you kind of imagine an animal moving in space as they're moving through a place field. And you get spikes, and this firing rate increases, more and more spikes as you get further into the receptive field. And he noticed that the first spikes here occur at around the peak. But as the animal moves further and further into the field, as the firing rate increases, the spikes occur earlier and earlier in phase, that there is an interaction between the phase of firing and the position of the animal. What he referred to as it was a phase coding of position. And this progressive advance in phase is a function of position. He termed phase precession, this advance. Now, just to illustrate, there's another thing here that I'll just point out. I'll get back to it again later. Now you say, oh, okay, yeah, the theta, this oscillation, there's some relationship between the phase of firing. But what else can these dynamic modes or oscillations do other than coordinate the timing of spikes within an area? They can also serve to couple or coordinate the interaction or communication between areas, and that is downstream areas. If you're going to listen to the spiking of the hippocampus, it's like, I want to know what the hippocampus is saying. Well, if the hippocampus only talks at the peaks of the theta oscillation, if you're downstream, you might want to know when the peak of the theta oscillation is, right? You might want to phase lock to remote structures. And what we see here is, this is actually another recording made in a remote area. This is the other top of the hierarchy executive area, the prefrontal cortex. And when you record simultaneously in the prefrontal cortex and the hippocampus, what you note the spiking of this neuron in the prefrontal cortex. It's actually phase locked to the hippocampal theta oscillation. And as the prefrontal cortex both knows and cares what this oscillation in the hippocampus is doing, which would make sense if the prefrontal cortex is listening to or interacting with the hippocampus. And I think I actually cut this out of the talk, but we did sort of a series of experiments demonstrating that this phase locking is actually indicative and sufficient to indicate when the hippocampus and prefrontal cortex are actually communicating. That it's not present all the time. It's only present when animals are actually making spatial decision. So it's like, this is the idea of dynamic functional coupling, that you could have two structures that talk to each other because they're actually wired up together. But you can also have structures that talk to each other when they need to talk to each other. And that's the idea of this dynamic phase coupling. So the hippocampus, prefrontal cortex are wired together, but they only talk to one another when they need to, and when they need to is indicated by this phase locking phenomenon. Question?
Sorry, I am so lost.
Let me try to help you find your way.
So the red line you're saying is spiking in response to the line below that's a black with the blue.
These are two different measurements. So this is the local field potential. So this is what the population is getting as kind of input. Yeah. This is the spiking of a single neuron in the hippocampus.
Okay.
This is the spiking of a single neuron in the prefrontal cortex.
Sure. So when you say phase, is that indicated by that dotted line?
Yep, exactly. That's kind of zero phase. That's the peak of the theta oscillation in the hippocampus.
Okay, and then the numbers that you have, the 1, 2, 1, 2, or
these are spikes. These are just like ticks. They're not.
Okay.
You're reading too much into there. You're trying to decode the, the spiking pattern. So this is like one spike. These are like two spikes that occur. So this is the occurrence of an action potential at that time.
Okay.
Right. And so here you see more spikes. Early on, you start with just like one spike as the animal enters the field, then you get two spikes, and then here like 5 spikes. That's what we're seeing.
Thank you.
All right. Once lost, now found. Great. So a couple of things here. There's this phase locking, hippocampus spiking to the local field. There's remote phase locking, spiking in another structure to the local field. But then you also notice, this is just kind of put up here. This is the local field in the prefrontal cortex, and you're like, where's the oscillation? Where's the local field? It just illustrates that you can get this kind of phase locking, the prefrontal cortex phase locking the hippocampus without it locking to the local oscillation. And what that tells you is that this field is generated by the inputs coming in from other structures, all the synaptic inputs. So what it's saying is, you know, prefrontal cortex is getting a lot of inputs, and not all of them are actually rhythmically modulated like the hippocampus. Some of them are and some of them are not. So you may not see that here, the inputs, you know, expressing this dynamic oscillation, even though the output can reflect it. You need to look at a remote structure, the one that's actually driving it. So again, there's this phenomenon that O'Keefe discovered, phase precession. And you say, man, Matt, you're kind of getting, you know, if you kind of lost me with the spiking and the oscillation, you're really going to lose me with this. But I'm going to try to illustrate this with this little cartoon. And so this is, on the right panel, this is real data. This is actual data that illustrates the phenomenon of phase precession. And what it is, so we've got on this axis the position of the animal. The animal's moving in space, like running from left to right. The place field is this region from like 210 to 280 centimeters. And each of these points is an action potential, spiking. But now we've also added, in addition, if I just like collapsed all this down and it was like the number of spikes as a function of position, right, it would form something like this. This is the place field. Firing goes up, firing comes down, right? But here we can add in the phase of firing with respect to this theta oscillation. And so what you see here, this is what phase precession is. As the animal enters the field, you get a couple of spikes, and this is averaged over, you know, over multiple trials. But on average, you're getting a couple of spikes here, 2 or 3 spikes, but when they occur late in the theta cycle. As the animal gets further and further into the field, the spikes occur earlier and earlier. So again, if I imagine as the animal's moving through the field, you get like first a spike, spike, you know, pop, 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 and the spikes fire earlier and earlier in phase, this relationship. Now here's a cartoon that I use to illustrate what I think is sort of a biophysically plausible explanation. How could you get something like this? Without, you know, requiring some, I don't know, elaborate coding scheme. It just says, look, this is just what cells do. And so the idea is simple, and that is that there are 2 interacting mechanisms in generating an action potential in a cell. And it's this excitatory-inhibitory interaction. You have excitation coming in through these synaptic inputs, and these synaptic inputs are going to be spatially biased, and that's shown in blue. In blue, as the animal moves through space, the inputs coming from like visual, spatial. Let's say I have an input coming into the cell that, let's say, associated with that chair. As I get closer to the chair, the input gets more, you know, is more pronounced. So I'm going to have spatially graded excitatory input. the same time, I'm going to have dynamically modulated inhibitory input. So inhibition is going to be changing rapidly, and typically the firing rate of inhibitory cells is much higher, and inhibitory cells track the theta oscillation. So if you record from inhibitory cells, their firing rate is going to follow the theta oscillation. So their inputs are likely to vary in this dynamic fashion. So you got spatial dynamics here in excitation, temporal dynamics here in inhibition. And then the biophysical model is very simple. It's just that you get a spike when excitation, blue, is higher than inhibition, red. That's it. So if we ask, okay, given that simple model, when would spikes be generated by this single cell? And so animals entering the field, excitatory input is weak. You have to wait till inhibition drops all the way to here before you get a spike, right? It's over here. As the excitation increases, you can fire earlier and earlier. I don't have to wait until inhibition drops this low because excitation is higher. And so what you're going to get is more excitation, earlier firing. It's just it's just an intensity to latency transformation, which you could do in a single cell. It doesn't require a network. It's just this is what cells do. Stronger input, earlier firing. And this simple biophysical model is going to give you a couple of other things that you note in this sort of phase precession. This is the real data. And that is that these spikes the first spike certainly the timing will be dependent upon the phase and the relative excitation. But the larger the excitation, it can fire basically any time blue is greater than red. Right? So you can potentially going to get more spiking. This is an increase in the firing rate. And more variability in phase. The spikes could occur anywhere in here. And that's exactly what you see here. And that is, as the number of spikes increases and the phase advances, the spread in this function increases. Phase variability increases. And I like to point out, if you kind of look at this and you said, oh, okay, this is an example of like temporal coding. And this is what O'Keefe originally advanced when he discovered this. He said, based on this, the real hippocampal code for place is not the firing rate, but rather the phase. And that's because if I just tell you that this cell fired, you can tell me that the cell is somewhere in its receptive field. But if I give you the phase, you can precisely position it late phase. Oh, I know exactly where the animal is. It's somewhere in this region. So the more precise spatial information comes from deciphering the phase. But I'm just going to ask you, normally I would do this, but I would sort of mask this. If you just look at this, there are actually really kind of 4 quadrants in this phase precession diagram. If you just look at the activity from 180 to 360 degrees, the late phase information, and you just look at the spiking, just ignore the bottom part, you see that this is actually, it's pretty good. It's pretty linear, what I was saying, the temporal code, phase and space is pretty good. If you look at the activity here, on the other hand, from 0 to 180, it's actually not that great, right? I mean, there's a lot of spikes, but, there's not really a good linearity to the phase rate, relationship. What that suggests is that there are really kind of 2 coding regimes here. Late phase, where you have temporal coding, and then the early phase, where you're getting a lot of spikes. So if I tell you, man, this hippocampal cell, it produced 10 spikes in this brief period of time. You say, oh, the high rate region, that's over here. I don't need the phase information. I just need the rate information. So I have temporal coding, rate coding. That's also the case if I separate it in this way. Early portion of the field, late portion of the field. The early portion of the field, again, good linearity. Late portion of the field, poor linearity, but good rate information. So this suggests that the receptive field actually has these 2 coding regimes, temporal coding and rate coding regime. I think that I probably cut out part of this talk where I kind of get into, is those, it looks like those are sort of meaningful, but can we actually demonstrate that those coding properties actually are functional? And we kind of used closed-loop phase-locked optogenetics to selectively inhibit firing at different phases of the theta oscillation. Say, okay, now I have a hippocampus that can only communicate either the early or the late phase information. What you find is when you restrict the kind of the firing to the early or late phase information, you get precisely this kind of temporal coding information. Animals use information about where they're going to go to, And when you restrict it to the rate coding, animals use information about where they are. You can see this in behavioral decision-making tasks. You can follow with me later. I think I cut that out, but but if we take that principle, the idea of these kind of two coding regimes, but also that phase is actually relevant to the coding, what does that actually, that's a single cell, what does that mean at a population level? And so to illustrate that, I showed this cartoon where now I have two place cells. They're both exhibiting phase precession. One place cell is before the other. So you imagine animals going through space, place cell 1 fires, then place cell 2 fires. There's a spatial sequence. And if I now look at the timing, what is the timing of the spikes that are emitted by these two cells? Place cell 1, because of this gradient, it's the intensity of the excitatory input to place cell 1 is always going to be higher than place cell 2. That's just because there are these two gradients, which means that the spikes from place cell 1 are always going to fire before place cell 2 on every theta cycle. And what that means is, as the animal moves through space, place cell 1 firing before place cell 2, on each and every theta cycle, place cell 1 will fire before place cell 2. There's a sequence, 1, 2, 1, 2, 1, 2, 1, 2, on every theta cycle, and that is as a representation of the spatial sequence translated into a temporal sequence within each and every theta cycle. And this does two things. One is it transforms the spatial time scale, or the sort of the spatial scale, spatial sequences into time, and it maps the behavioral time scale. How long does it take to get from, this location to that location? It depends. The animal, if he's moving fast, it's one time. If he's moving slowly, it's another time. But if the intent is to simply capture place cell that, animal goes through location 1 followed by location 2. If you simply want to capture temporal order rather than absolute time, this does that. It transforms absolute time into relative time, temporal order, and it maps it into a fixed biophysical time scale, about 100 milliseconds. Now, does this actually occur? Well, so if we take that same approach that I showed you earlier, this is decoding, this is using Bayesian decoding, but now we're just doing it at a very fine temporal resolution. Previously, I was showing the little, the movie with little triangles. It's like every 200 milliseconds, we're asking which cells are firing, decoding position. Here, we're doing it at 20 milliseconds. So every 20 milliseconds, the theta rhythm is about a 10 hertz rhythm, which means it's about 100 to 120 milliseconds. That's a period. And so what we're looking at here is we're able to decode at a resolution that's finer than the time scale of a theta oscillation, sub-theta, sort of wavelength decoding. And so what you see here, if you decode at this finer resolution, in the dotted line, you see this is the actual trajectory the animal is moving. This is in time, and this is in space along that track. So what you see now, this is just over the course of about a second and a half. What you see is that the code, and you see these little kind of sweeps here. The hippocampus is not just coding where the animal is. There are these little sweeps that seem to go from just behind the animal, this is behind the animal, to just in front of the animal. And that these sweeps are locked to the theta oscillation. This is the theta oscillation that's going on at the same time. And we just kind of average these things. So we take, the sort of the phase-locked theta oscillation and ask sort of what is the spatial code relative to the animal's current position on each and every theta cycle, pool them all together. That's what we get here. So this is the actual hippocampal code at the highest resolution relative to the theta oscillation. What you see is that the theta code, that the hippocampal code sweeps from just behind the animal to just in front of the animal. It does so every, 10 times a second. So when the animal is moving through space, the hippocampus is not saying, oh, you're here, you're here, you're here. It's saying, you came from here and you're going there. The sweep, 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 sweep. Behind, ahead, past, future. And it turns out that these different components, the future component is captured in this late theta phase, the part of the theta oscillation where you're getting the temporal code. So you're kind of going from rate coding, tell you, okay, this is where you just came from and where you are, with a firing rate that can inform that with some degree of confidence. So you can actually use that intensity of the firing rate to say, I'm pretty sure I'm at this location based on my population firing rate, but where am I going to go? Where am I going to end up in this trajectory which at typical velocities is like, where am I going to be in the next 1 to 2 seconds, 1 to 3 seconds. So I'm kind of estimating my future position a couple of seconds into the future. And now I'm using this temporal coded region to get an actual trajectory or spatial sequence to do that. So now we have this idea, okay, the hippocampus can capture temporal sequential information using this rhythmic strategy, theta coding, temporal coding strategy, but is like this temporal sequence information actually used to process memory? What is memory? I haven't showed you anything about memory. It's just like, okay, this is just what happens. So memory is minimally, it's like the retention and re-expression of like prior information. Something's retained and I can get a readout of it later. And the idea that this kind of readout might occur during sleep is actually useful because if you do see something during sleep, because the animal is kind of disconnected from the outside world, this information must be generated internally, and it must arise from some kind of retained information. So by definition, it must be either the consequence of or a reflection of memory. And we kind of identify these two states. How are we doing in time? Yeah. I'll speed it up. These kind of two states, two stages of sleep, referred to as non-REM and REM sleep. REM sleep is what we typically think of as dream sleep. We call it paradoxical sleep because if you actually record brain activity, look at brain rhythms, it looks just like the animal's awake. And the only difference is that you have your motor neurons are inhibited, so you can't actually move. So you're kind of paralyzed. It's like you're awake. Your brain looks like it's awake, but you can't move. Non-REM sleep is this evolutionarily older. It looks very much like quiet wakefulness. In fact, the non-REM state has even been argued to be manifest in invertebrates. There are these sort of fly models of sleep that are essentially periods of immobility. As I pointed out, immobility and non-REM sleep in the hippocampus look essentially indistinguishable. So there's this idea that non-REM is kind of like this extended state of immobility, evolutionarily conserved. What goes on during these two states, or these two stages of sleep? I won't talk about REM. In REM, I can simply describe that we can decode activity during REM, and what you see is this kind of recapitulation, replaying of an animal's experience, essentially at what I refer to as real time. So it looks very much, if you decode activity when an animal's in REM, it looks like the animal's running. In fact, that's how I sort of first stumbled across sleep memory reactivation. I was actually I had an animal that had just run. I was still recording from this. I took the animal off. I went to, do some work, and I was listening to brain activity, and suddenly I heard something that sounded like the animal was running around, and I was worried that the animal maybe had jumped out of the sleep box. And when I turned and I saw the animal was just sitting, it's curled up, and you see the little fictive movement, and it was in REM sleep. So REM sleep is like, wow, sounds like the animal's awake. I should look at this activity. But it turned out that where, a lot of the interesting memory-related activity was expressed was in this other stage, non-REM sleep. What does non-REM sleep look like? And so here, this is just like a simple experiment, kind of illustrating the phenomena. So an animal goes to sleep, we run him, and then put him to sleep again. And then here you see this is real sleep architecture in gray, or just turned out to be green for some reason. This is periods of non-REM sleep. In red are periods of REM. And so sleep architecture, periods of sleep onset are going to be associated with non-REM. So when you first go to sleep, you're going to first fall into light sleep, so-called these non-REM stages, non-REM 1, then it evolves in non-REM 2. It's all non-REM sleep. And then as you get deeper and deeper into sleep, at some point you'll make a transition into REM, that's in red. And then back into non-REM, then REM. And then over the course of a night, of a larger sleep cycle, the frequency of REM will increase. So what you're seeing here, at the end, the animal's just about to wake up. This would be 6 or 7 in the morning when you're just waking up. You're getting longer and more intense REM episodes. In fact, it's likely that the last sleep stage that you would experience before awakening would be a REM state. Because, after you come out of REM, there's sort of a bifurcation. You either go back into non-REM sleep or you wake up. And so you can kind of think of there's this alternation of non-REM and REM. If you think about it, and I didn't illustrate it here, REM is indicated by the presence of these rhythms, the theta rhythm. When I say brain activity looks like it's awake, It looks like it's awake because the rhythms that are associated with wakefulness are expressed. In fact, the most intense theta oscillations that you see in the brain are during REM sleep. And that these things are actually controlled by neuromodulators, including acetylcholine, which is one of these primary neuromodulators, low acetylcholine associated with non-REM, high acetylcholine associated with REM and wakefulness. And again, the highest acetylcholine levels are associated with REM sleep. So REM is like supercharged wakefulness. If you look during wakefulness, you're getting basically the same thing, alternation between these theta-like states, awake-like states, and then these quiet wakeful states, which look like non-REM. So you can think of this, sure, there's like awake sleep, but you can also think more broadly, actually, there's just this alternation between REM and like theta and non-theta, online and offline. It's just that the online state during wakefulness is being driven by information coming from the outside world. The theta state driven during sleep is being generated by the internal world, and that is that there's that cortical areas that are responsible for perception and action are now driving the hippocampus in a way. It's like synthetic experience. And so you can say this is the evolutionary adaptation, that this basic switch between, let's say, wakeful and quiet wakeful activity, this is just part of the biology. And then for some species, there was a need to conform to like circadian regulation of behavior. Things are dark. If you're, largely driven by sort of, let's say, visual perception that's not available, and you want to have some downtime, you go to sleep. But you still want to engage in some sort of useful, constructive cognitive processing. And so you create this synthetic REM state and alternate it with non-REM. And so there's this idea that this alternation, what goes on in non-REM, and so non-REM REM is like quiet wakeful, active wakefulness. So what goes on in these non-REM-like states? I gotta really speed up. And just to sort of briefly point this out, when the animal goes to sleep, you see these sequences when the animal runs. This is like this theta sequence stuff, but there are these spatial sequences that get recapitulated. You see them reappear, and that is when the animal's asleep, you see these sequences reappear, and these sequences actually are expressed during these sharp wave ripple events. I've taken the sharp wave away from you. This just shows you the ripple. But you get these compressed spatial sequences. So a sequence that may have taken the animal a couple of seconds to run through when it was awake get expressed over a couple of 100 milliseconds. And that if you record outside of the hippocampus, for instance, the visual cortex, so we're going to simultaneously record in the hippocampus visual cortex, you find that you get spatial sequences in the hippocampus, the hippocampus, and in the visual cortex. So there's like a spatial correlate in the visual cortex as well, and you get spatial sequences in the hippocampus, spatial sequences, or like visual sequences in the visual cortex re-expressed during non-REM sleep. And the two are coordinated. And so it's the idea that during non-REM sleep, hippocampus is reactivating sequential memory in a compressed form. The neocortex is responding to that by expressing the corresponding sensory sequences. You can think of this as something like the analog of rat dreaming. There's a perceptual re-expression of some kind of experiential state with the perceptual components attached. And that these things are actually tied to sort of a ubiquitous property of this non-REM state, and that is that it's indicated by the presence of these slower oscillations, the low-frequency oscillations, termed slow waves, as you can see in this is the EEG, or local field potential, recorded at the surface, and that if you record activity of individual neurons here in the visual cortex and in the hippocampus, what you see is it seems to alternate between these periods of elevated activity, these so-called up states when cells are active, interleave with these down states. And so there's a slow oscillation that's also associated with this alternation of active, inactive. And activity in the neocortex is sort of locked to activity, these up-down state transitions in the hippocampus. You kind of note that there seems to be a relationship here where the neocortex turns on, and a little bit later the hippocampus turns on, neocortex, hippocampus, suggesting that there's some kind of dialogue. neocortex driving the hippocampus expressing sequences that are tied to the expression of sharp wave ripples, and then those sequences go back to the neocortex. So there's some kind of sleep processing associated with slow oscillations, reactivation of these sequences. And so if that's the case, and that is that when the neocortex turns on, sensory cortex turns on, it drives the hippocampus, can we drive the sensory cortex? Now, this isn't visual cortex, but the auditory cortex is interesting because it's accessible during sleep. As you hear, when you're asleep, you hear everything. You hear just as well. The auditory cortex is just as responsive during sleep as it is during wakefulness. The only difference is that your arousal levels, arousal being response to stimuli, is diminished. You're less likely to respond to it or wake up. So you can pump in sounds when you're asleep and drive sensory cortex, which means if we delivered sounds here during slow wave sleep, we should be able to influence the activity of these sensory cortical neurons. And so we did this experiment. Okay, so we'll have a task where the animal is using sound to kind of direct its spatial behavior. You hear one sound, you go right. You hear a different sound, you go left. Animal goes to sleep, and you just play these sounds, the left associated sound, the right associated sound. And just in summary, when you do this, so here's the animal actually running the task, position, time, animal runs left, you get some place fields on the, or animal goes to the right, place cells on the right, animal goes to the left, you get place cells on the left. Here are the different place cells. And now we're just going to deliver the sounds associated with right and left while the animal is asleep. So now animal goes to sleep, you're getting activity, and then just going to start playing sounds. So here's an example, play a sound for a fraction of a second, and then record the hippocampal reactivated memory patterns that follow from that over the sort of the following 5 to 10 seconds. And what you find is when you play the left-hand sound, when the hippocampus reactivates memory, it tends to be memory for the left-hand side of the maze. So you can trigger and influence and control the content of hippocampal memory by biasing the activity of sensory neocortex during slow wave oscillations. And this is just the data. It's published, so you can look at it if you want. But that was the basic result. And a couple of things that are interesting here, and that is sound is delivered here for just 750 milliseconds. The reactivation bias persists for 5 to 10 seconds. In fact, if anything, this is the relative bias. First 5 seconds, second 5 seconds. If anything, the bias gets stronger. And it suggests that you're kind of triggering, you're not driving hippocampal reactivation with the sound. The sound is simply establishing an initial condition, a bias that is propagated and even amplified through this dialogue. And so it's this idea that there is this internal dialogue that is driven by the interplay between, okay, this is what I heard, this is what I remember. Well, based on what I remember, what do you think I heard? And so you kind of think of this this kind of recurrent process of both evaluating, generating, and then reevaluating information going on during sleep. Now, this can be demonstrated empirically if you actually do this in humans, and there was a really cool study, I'm just going to kind of point to it here, in Ken Paler's lab at Northwestern University, where he actually did this experiment, a psychophysical experiment, where he had people performing this little simple task like this game of concentration, where you have to remember, you have these little cards, you have to remember where the cards are. The cat card is down here, the teapot card is up here. You just have to kind of remember, it's a sort of a visual spatial memory task. But what Rudolph and Paler did was, in addition to showing the image, they played a sound. So you flip over the cat, you see the cat, you hear a cat sound. And then during sleep, and in particular during non-REM sleep, they would either play like the cat sound or the teapot sound. And when they played like the cat sound, people wake up and they do the task, and lo and behold, they remember the cat, where the cats are better than the teapots. And that is, there has not been, not just sleep-dependent memory enhancement, but has been target-specific, stimulus-specific enhancement. They remembered the thing that was actually biased or triggered during sleep. And it occurred with like cue delivery during non-REM sleep. I have 3 minutes? Yeah, all right. Nah, I don't want to take too much more. So this is what's going on during sleep. As I pointed out, these kind of sharp wave ripple events during non-REM sleep look very much like quiet wakefulness. So let's look at quiet wakefulness. What goes on during quiet wakefulness? It looks like non-REM. And it occurs when animals stop or are immobile and are engaged in what's referred to as consummatory behavior. Anything that involves, like, you're just satisfying internal needs. And so that can include things like grooming or eating. So eating food reward. So in a task like this, animal runs down a track, eats some food reward. While it's eating, you're getting these sharp wave ripple events. And so here's a task involving, it's just like a longer track. Animal runs from one end to the other. It's the one I showed you in the movie. And in fact, I'm going to kind of zoom in on a single trial. The animal walks from one location on this 10-meter track, gets to the end, and then just sits there and stops. And while it stops, it's consuming the reward. Now you start to see that activity, these are the sharp wave ripples. This is what the spiking activity, so you see these bursts of spiking. And now we're going to kind of zoom in on this little one event. And this one event looks like this. Now, one thing I'll point out is, so you see here like this, it's periods that's like quiet, not much spiking, then a whole bunch of spiking, and then it's quiet. Looks a lot like that upstate that I showed you in non-REM sleep, right? It looks like non-REM sleep, except it's like one cycle, one slow oscillation cycle of slow wave sleep. So the thing that might distinguish sleep and non-REM sleep from quiet wakefulness is non-REM sleep is you just get a lot more of these things, right? You're able to engage in a dialogue with the neocortex. Whereas in quiet wakefulness, it's very much, okay, I got like one upstate. I'm going to be influenced by the current sensory information, perhaps from the cortex. So it's kind of like the one-shot version of non-REM sleep. If we zoom into this, this is the sharp wave ripples. And in this case, this is about 500 milliseconds. A sharp wave ripple event is about 100 milliseconds. And so what you get over one of these upstate-like periods is actually multiple sharp wave ripples. You get a burst of them. You get, like, in this case, there's like 4 of them. And now I'm going to decode that activity. This is the decoded activity during one of these upstate-like periods with multiple ripples. And you see the estimated decoded position goes from this location to this location. It's a sequence, it's a trajectory. So during this slow wave sleep, you're seeing, just as I illustrated earlier with the spiking, this is now a population decoding. You're getting reactivation of a sequence. And that these sequences slow down. This is what it looks like here. Animals stop, slow down by about a factor of 5. It's like expressing this extended sequence. And one thing about this sequence is that is an example of so-called forward replay. Here's another example. Animal stopped here, and what you see is that that sequence can be played in reverse. It's actually going backward. So during stopping, quiet wakefulness, you get both forward and reverse reactivation of extended sequences. And that these sequences, when they're expressed, longer sequences are expressed over multiple sharp wave ripples. So the idea is that there's like a quantum of experience, about 100 milliseconds in duration, and when you extend that, you try to, animals like replaying or thinking about extended sequences, you just kind of link those together in these chains of sharp wave ripples. And I'm just going to point out that, and there's like a characteristic speed or velocity. It's about 8 to 10 times actual behavioral speed, and that the individual chunks, so those sharp wave ripple events, about 100 milliseconds in duration, well, that's exactly the duration of a theta cycle. Theta cycle is about 100 milliseconds, the period. And if you actually look at the reconstructed, this little sequence here, it's on the same spatial and temporal scale as the sharp wave ripple. So the idea is that information is encoded, captured in these small sequential chunks. The sharp wave ripple I referred to as like a compressed replay. It's actually not a compressed replay. The compression occurs during encoding. And then you just take multiple theta cycles, these theta sequences, and you chain them together. And so that's essentially memory is constructed out of these little theta sequence chunks. And that these chunks, the question of what would you use these replayed sequences for? And there are 2 really functional hypotheses. One is, well, what's the animal doing? It's like I saw the forward sequences. That's kind of like planning, right? You're sort of thinking forward. I'm here. Maybe I want to get to the end. Well, David Foster, who initially discovered this reverse replay, and thought, well, okay, if this is planning, if we put an animal in a task where it's here, it has to try to find a hidden location, and we look at these replay events, do they seem to reflect, anticipated future goals? And what he found is, yeah, they do. So the animals replaying these sequences, they sort of tend to go in the direction of the goal where the animal wants to go to. But there's another model, which is to say, well, what about those reverse sequences? Why would you play things in reverse? It's not like, oh, I want to learn how to not go where I was. Reverse reactivation can be used to solve a problem in general, sort of what we refer to as kind of unsupervised learning, where the animal, it's like trial and error learning, where you're not told how to do something. You only learn, about whether an action or set of actions was appropriate based on the reward after it's occurred. And so the problem with that is when you do something, you say, oh, that was great. What you need to know is not, that was great. It's like, what did I just do? How do I reinforce what I did in the past in order to get this unexpected reward in the future? And that's referred to as the temporal credit assignment problem. How do I give credit to things that led up to present reward? And one way to solve the temporal credit assignment problem is to take that event, so the animal walks to the end, it gets reward here. If you could spread that reward backward in time, say, oh, spread it backward in time, and then associate it with the reverse sequence, you could solve the temporal credit assignment in one shot. So that's the idea. So maybe in reinforcement learning, solving the temporal credit assignment problem, maybe that's what this reverse replay is associated with. Well, if that were the case, and you actually looked in the reward areas, you should see a relationship between replay, maybe both forward and reverse, because they both involve reward, and reward signaling. And I'm kind of over time, so I'll just kind of give you the short, sort of the short answer and version of that. And the answer is that indeed, when you record in reward areas, record in the VTA that provides, dopaminergic innervation to large areas of the forebrain, including the hippocampus. Record in these areas in a task. Just going to sort of get to the punchline here. What you find is that you have dopamine activity that is locked to sharp wave ripple events. So when the hippocampus produces a burst, yep, dopamine cells are firing, and that's just shown here. This is firing of dopamine units with respect to these hippocampal sharp wave ripples. Hippocampal activity locked to sharp wave ripples, dopamine activity locked to sharp wave ripples. And if you decode the activity in the hippocampus, say, okay, it occurs during sharp wave ripples, but what about within a sharp wave ripple? Now we're going to go even finer, right? Say we're going to decode individual positions in one of these trajectories. Individual trajectories, let's, for example, and ask, where does this, where does this stuff actually fire? And so this is now dopamine neuron firing as a function of replayed position within one of these events. And what you see is that the dopamine neurons fire when the replayed trajectory lands on a goal location. So basically the animal is thinking or imagining of running to the goal location. You get dopamine activity reinforced. So it's okay, that's what you would imagine if there really was this tight coupling. Now what was interesting is when we did this is all during quiet wakefulness. And you might think, oh, sleep is like non-REM, right? It's the same thing. You might expect to see the same thing. Sleep there's learning, right? So you say, oh, so that must be going on during sleep as well. When you look at the relationship between, this is dopamine activity, sharp wave ripples during quiet wakefulness, dopamine activity with respect to sharp wave ripples in sleep, you don't see it. Dopamine activity is not coupled with reactivation during sleep. It's like, whoa, wait a minute. So the hippocampus is doing the same thing. The dopamine system is not doing the same thing, but I thought learning and memory, it's like reinforcement learning, right? Isn't that the idea? So maybe the dopamine system just doesn't work during sleep, right? Maybe there's something, it's like plasticity rules don't work during sleep. Well, there was a very interesting experiment done by Karim Ben-Shanain in Paris trying to address that question. What he did was closed-loop stimulation of this VTA dopamine projection. So he activated the reward system when an animal was asleep, and he made that activation contingent upon a place cell. So when a place cell fired when the animal was in non-REM sleep, trigger reward. And place cells would fire in a certain, so here's a place cell, for instance, that would fire in this part of the maze. what he was able to do was he was able to implant a new memory, and that is animal explores a box, like, okay, box is a box, right? And then he ties reward to the activation of a place cell in this location, brings the animal out, and all of a sudden the animal's like, I don't know, this box, but I really like this location. It's an acquired place preference, completely de novo memory. The animal had never experienced the pairing of location and reward while it was awake, only when it was asleep, and it expressed it. So what this says is, man, dopamine-driven, reinforcement-driven learning can be very powerful during sleep. You turn on dopamine during sleep, the stuff you dream about, you will actually now express during behavior. Now, I don't know about you, but I certainly wouldn't want to be doing what I dreamt about when I was awake. And so the idea that there's a different function to the processing of memory during sleep. And you say, what could that be? What kind of memory processing could you, useful memory processing, could you engage in during sleep that didn't involve reward? And I'm just going to kind of point to, there's some, I'm not going to have time to go into it in real detail, but there was this whole class of learning that really drove this so-called cognitive revolution. And it was the observation, largely driven by the work of Edward Tolman at Berkeley, identifying a kind of a form of learning that was not driven by behavior. Before this, it was like the behaviorists, there was like the Skinner boxes, like I can teach you to become a fireman by just like giving you cookies every time you do something related to, what you need to do, to be a fireman. I mean, so it's all learning is basically reward contingent, association. Tolman demonstrated animals learn things just by being exposed to environments, so-called latent learning. No reward, just exploration. And it was the idea that kind of learning could result in the formation of like a cognitive map that you could use to do planning on. So there's a form of that you learn representations that make other tasks learnable. So there's both learning and there's learnability. So constructing useful representations is kind of a critical part of learning, and that doesn't require reinforcement. And so, postdoc in the lab using calcium imaging explored this idea. And so just recorded in animals, populations, animals exploring in a maze. I promise you I'm almost at the end now. I'm just going to kind of jump to the end. We're going to do the same idea, record a bunch of neurons, only this is using calcium imaging. But now the idea is I'm going to take these neural populations, and instead of decoding actual position, which I showed you how to do, I'm going to say, can we just use these sort of manifold learning approaches, dimensional reduction approaches, to ask what is the abstract space that might capture the kind of correlated activity among these populations. In other words, I'm going to use manifold learning to construct spaces which capture the structure of this activity. And so there's going to be this sort of mental map. This is like the cognitive map. There's the actual map where the animal is, and then this is the mental map that reflects the relationships between the firing of neurons. And you can kind of see this if I'll just play this little movie. And so this is illustrating, this is where the animal is actually going here, and we're kind of decoding location on the actual apparatus. And this is the decoded location on this mental map. So we create a manifold and we map ongoing activities the animal's running onto this mental map. All right? And now we're going to say, okay, so we got a physical map. This is the location of the actual location of the animal, and this is the location in this mental manifold space, mental map space. And the question is, what happens when you just expose an animal to this space? Does the mental map change? And so here you can see an example. Okay, the first time you put it in here, and so this is the physical map, it's a little T-maze. This is like a little square maze. And you see after 5 sessions, and now we look at the basic sort of, the topology of the mental map, and you can see with experience, it seems to conform more closely to the actual physical map. The same thing here. It kind of starts like this, and then it ends with something that looks is more similar to the mental map. And this changes, the similarity changes with experience. But the interesting part of this is that when you break this down, so the animal's going to all right. So the technology has said, you're done, Matt. You're done, man, because we're not going to show you any more slides. Oh, there it is. When you break this down, say, okay, now we're going to just like sing adjacent sessions. So animal runs in one, sleeps, and he runs in another one. Okay, that's like kind of weird. That's a weird way of like, it's kind of like the hook, like drawing you off. Okay, show you the slide. You can't actually. If you look at the change that occurs is over sleep. So during sleep. So if you with intervening sleep, that's where the similarity goes up. So if you look at the map before and after sleep, it's during sleep is what actually changes it. Sleep changes the representation of the mental map to conform more closely to the physical map. And then there's some other stuff about, that when we actually look at the changes, it's not all cells that change. It's just some. In fact, there are those that are kind of strongly spatial that represent the location. Think of like this kind of rate coding. And then there are cells that evolve, that develop with additional experience, weakly spatial, but they become more correlated. And the speculation is that these are cells they're kind of cells that they're kind of the strongly spatial. And then there are kind of cells that serve to link the spatial cells. You kind of think of that as like what changes with experience. It's not the locations, but the way you explore those experiences. So kind of think about like the trajectories, the temporal structure, the actual experience within an environment is something that evolves with experience. And from that experience, you're able to create a mental map that captures the topology of space. And topology of space is not just where things are, but how they're kind of connected in time as I explore them. So as we learn to think about how you move through space, that's what goes on during sleep. And during sleep, that's what we see, expression of these trajectories or temporal sequences. And for this, you don't need reward because it's not about getting anything. It's about understanding how to create representations upon which you can then apply this kind of reinforcement-driven learning. All right, that's it. I think that's it.